which are moved in extension, which is on two important, clinically important enzymes, lactate, dehydrogenase and cyclooxygenase. I invite the speaker, Dr. Andrew. Welcome, sir, and over to you.
Thank you. I would like to get your attention. I don't know whether you are, active, but when I speak, I want my hearers to be engaged and to understand what I'm saying. So, at the outset, I would like to thank the organizers at CMFRI, especially Professor Murli, for the opportunity to discuss the work that we did jointly on two important enzymes in human physiology and medicine. One is lactate dehydrogenase and the other one is cyclooxygenase. So I go without much ado into my talk. Next slide. Good here? Okay. Fine. So I'm going to discuss these two aspects from published papers that we have. They are hypothesis papers. We are drawing on lactate dehydrogenase. We published this work in the Journal of Cellular Physiology, Wiley, where we said there are Morburn precepts for lactic acidosis, Cory's cycle, and Warburg effect. And we talked about how dehydrogenases, protons and oxygen have interactive equilibrium. Now, lactate dehydrogenase is an enzyme which reversibly, according to textbooks, it reversibly converts lactic acid or lactate to pyruvate. And it is a oxidoreductase class 1 enzyme which utilizes nicotinamide adenine dinucleotide, NAD coenzyme. So you can see that active LDH is a tetramer. You can see that in the image, and then there is a NADH binding pocket, about 10 angstrom deep pocket there is, which stabilizes negatively charged lactate and pyruvate because there are positively charged amino acid residues within the enzyme active site pocket. So here you have the NADH binding domain as well as the substrate binding domain. So you can see.
Look at your screen. Is there a problem on my end or your screen is not displayed?
Is the muscle or liver LDH. LDH-B tetramer, B4, is found in heart tissue and sometimes in RBCs. Now, you have got what is known as Cory's cycle. In the textbook, you know that lactate can be converted to pyruvate in the liver, but lactate dehydrogenase is supposed to be a reversible enzyme. The same isozyme, you can see colored in blue, for LDH-A4 or M or LDH5 isozyme is present in both liver and muscle cell. But then suddenly we had the question, if like in textbooks it is a reversible reaction, why should lactate, which is generated in the muscle, go to the liver and be reconverted to pyruvate and then to glucose through gluconeogenesis, and that glucose comes back and this forms a cycle? Why should that happen? It is a reversible enzyme in textbooks. Furthermore, we found that muscle has 4 times more LDH than the liver. So it makes no sense why it should go to the liver. So with that, we started questioning how LDH is involved in both Warburg effect and in Cory's cycle, and if there is an alternate explanation for why. The main question you understand is, why does lactate have to go to the liver? So I will show you how we solve this issue. Okay, now it's working. So you see that lactate dehydrogenase, as the name suggests, it is dehydrogenating lactate. That is, lactate to pyruvate is so-called forward reaction. That should be the forward reaction. The name of the enzyme is tied to the name of the substrate it catalyzes. So, LDH is a reversible enzyme according to textbooks with a delta G dash naught of negative 25.1 kilojoule per mole. And under physiological conditions, this should be something that the cell can, reverse. Right? This is the textbook calculation from Leninger. Okay, the equilibrium constant is like 10 power 4 to 10 to the power of 12. That is the range. But you can see here in this image, there are both steps 1 through 5, which show the backward reaction, right, of pyruvate to lactate, and the forward reaction of 5, 4, 3, 2, 1. If you go in that sequence where the forward reaction is lactate is reconverted to pyruvate. The Kd values are given there for you to see. The Michaelis-Menten constant for both these is given, and the equilibrium constant is supposed to be approximately 1. It can be about 10 to the power 4 to 10 to the power of 12. So I don't want to go more into this. Now, main question. There are two isozymes, LDHA and LDHB. LDHB is supposed to be mainly in the heart, and it converts lactate to pyruvate. LDHA is supposed to be in the liver or the muscle, and it converts pyruvate to lactate, according to scientists who have worked on this problem. And what is the mechanism of LDH action? It is acid-base catalysis. There is a direct hydride ion transfer which occurs within the active site, and that is how pyruvate becomes lactate. And again, lactate becomes pyruvate. It's all through acid-base catalysis because of histidine, and you have arginine, which is stabilizing the negatively charged ion of lactate or pyruvate. But if you look at the active sites of both enzymes, what do you see? They are superimposable almost. The position of NADH that you have, the position of oxamic acid, which is an inhibitor analog of lactate and pyruvate that you can see. The amino acid residues are all identical. Now, how does this differential recognition of lactate by heart LDH and pyruvate by liver LDH occur? There is no sense of that. There is no logic for that differentiation to happen. So the difference in topography or amino acid residues fails because the binding logic fails. Now if you see the LDH can, according to the classical cycle, it is a very highly ordered thing. NADH must bind first. NADH or NAD plus must bind first. Only then lactate or pyruvate has to squirm past that and go in and be bound to the arginine residues. So that is a very, very, very ordered way in which it happens, right? So it's step by step by step. This binds, then that binds, then there is hydride ion transfer. But in the Murburn forward cycle, what we can observe, what we can say is that ROS is usually generated in cellular systems, right? So when ROS is there, the positively charged residues in the active site can stabilize the ROS. So ROS can bind first. Sometimes NAD can bind first. So anything can happen. It's a reaction which is not, very highly ordered. So you have D-ROS. ROS can attack the lactate, which is bound somewhere on the surface. So in our docking studies, with blind docking, we found that lactate is binding somewhere else instead of the active site pocket in a more favorable way. So this modified or Murburn forward cycle shows that lactate can bind somewhere instead of the active site and still be attacked by ROS. ROS is what converts lactate to pyruvate, it is not LDH. LDH is merely facilitating that by stabilizing the ROS. And ROS can have more prolonged lifetime. That is why we can say that LDH can be called a Murzan. So, now, also singlet oxygen has been, in photodynamic therapy and in photosynthesis and many other systems, triplet ground state oxygen is converted to a very reactive form known as singlet oxygen. That is also seen in our system, right? So that can also be involved in, attacking the lactate. So there are very different ways. It's not highly ordered. So both the forward and reaction and the reverse reaction are altered from the classical pathway. You have more stochastic bimolecular interactions in the Murman purview, and DROS dynamics is there because of positively charged residues which can stabilize it. So we docked also the nicotinamide because we were interested in seeing what would happen. So now if you see lactate being converted to pyruvate, NAD plus should become NADH and H plus. So NADH is the product. If you see pyruvate becoming lactate, NADH is the substrate and NAD plus is the product. So we interchanged the NADH and NAD plus with both LDHA and LDHB. And we found out that the orientation of NADH can change. Right? So instead of the phosphate groups going in first, sometimes, instead of nicotinamide going in first, which should be closer to lactate or pyruvate, which is inside the active site pocket, it is binding in the opposite orientation with the nicotinamide ring coming out. And we found that this was more favorable energetically. So these kind of things suggest that, the system cannot clearly define the substrates, and that, binding can occur anywhere on the surface. Now you have a diverse molecules which are there which can serve as inhibitors of lactate dehydrogenase. Most of them are antioxidants, very surprisingly. I do a lot of work on antioxidants. You have gossypol, you have so many other things like galloflavin, sesamol, all are antioxidants. How do these antioxidants mess with the system? Do they block the active site channel where nicotinamide, cofactors, coenzymes enter and where lactate or pyruvate enter? And is that mouth being blocked and that is why we get inhibition? That is a question. But lo and behold, we even docked all those. So this is a diagram which can explain to you either with LDHB, where you have the forward reaction, that is lactate to pyruvate, or the backward reaction of pyruvate to lactate in LDHA, which is your liver and muscle enzyme. There can be different kinds of mechanisms that blockage can occur. An interfacial inhibitor can bind between the two monomers and give you lesser access to the active site channel, and that can be leading to inhibition. You can also have crypt blockers or oxamic acid, for example, which is known to go all the way in. So we have a lot of non-steroidal anti-inflammatory drugs which are, known to be either selective. There are also compounds which can go and bind there, and we also have LDH inhibitors. I showed you those inhibitors like gossipol and CDMIM, QABA. I'll tell you about all these things. EGCG is a very famous phenolic compound, epigallocatechin gallate. All these are known to go and inhibit the LDH system. Now the clearest question we can ask is, why do they inhibit? Because they are very good antioxidants. Are they inhibiting LDH because of binding, or is it because of their redox activity? That is the key question. Now we took structures of various LDH A and B from different mammalian sources. Some active site channels were closed, some of them were open. Maybe you can say it is protein dynamics. But there is a lot of difference in the sequence also, right? When you align them and check, all of them are not similar. You can see that binding of these things, gossipol and all these drugs happened at other places rather than the channel where you are supposed to see the binding in blind docking. Why did that happen? That's another question. So is it that there is dynamism in the protein, or is it like ROS is being scavenged by these molecules? That is the question. So now, the clearest thing is, why liver? Why should it go to the liver? The liver, according to the Morvan purview, is red because of high presence of cytochrome P450. Wherever cytochrome P450s are, where CPR is, the reductase enzyme, these enzymes produce a copious amount of ROS from oxygen. Oxygen is activated by these enzymes and ROS is released. So, we found out that, I'll show you the other slide first. So, when you have lactate, right, pyruvate and NADH plus H plus, you get lactate plus NAD plus. So, if you see this equation, the energetics of this equation was miscalculated in textbooks. We rightly found out that the role of the proton H plus and its contribution had been ignored. When you put that into the equation, you get a value, delta G of minus 476.4 kilojoules per mole. But if you go from lactate to pyruvate, you are reversing the reaction, the sign will revert, you will have plus 476. So the equilibrium constant is highly favorable, 10 to the power of 83 if you take the steady state concentrations of the lactate and pyruvate and the NADH and NAD plus, all these things, right? So you find out the equilibrium constant, it was about 10 to the power of 83, which means that this enzyme is going only one way. It is going from pyruvate to lactate and not vice versa. Because you need plus 476 kilojoule per mole, which is practically impossible to reverse even in a physiological system. But in the Morvan purview, because of these puppies of CPR and CYP, you see that their ability to produce a lot of ROS and the ability of drug molecules also to be there, right? Drug molecules are converted by this. You just had a talk on cytochrome P450 by my friend Abhinav. Now you see the contribution where lactate along with RH is a drug molecule, can be metabolized along with the drug molecule by the cytochrome P450 produced DROS, and the equation, the delta G that you have is negative 375 kilojoule per mole and the equilibrium constant is like 10 to the power of 67, which approaches that what you saw, the impossible thing is possible now because in the liver, along with drug metabolism and endobiotic metabolism also of our own compounds which we have, the liver is able to do this. That is why lactate goes to the liver to be reconverted to pyruvate, because it is a ROS rich place. Inside the cells itself, lactate goes into mitochondria. Now, if you see the cellular ratio of lactate and pyruvate, it is like 10:1. Normal condition, it is 10:1. That is why we can say that there is a 11th step in glycolysis, that is pyruvate becomes lactate. So, lactate is produced very high, 10 times more than pyruvate. But in cancer, the ratio of lactate and pyruvate can be much higher, like even 100:1. Why does that happen? We have an explanation for that. So, in the normal cells, on the left side, you can see that there is conversion of glucose to pyruvate and then to lactate. I told you lactate is the final step of glycolysis. The final product is not pyruvate, it is lactate. Now you have certain things here where there can be some Morvan in the mitochondria. lactate is made, but it can be utilized as a fuel because it is electron rich when compared to pyruvate. There are electrons going from NADH to make lactate, right? So compared to pyruvate, it is electron rich. But what is the problem you have is the redox recycling is limited because you also have end product inhibitions in the normal cells, which are not cancerous. In cancerous cells, you might think cancerous cells are not, just metabolically defective. They are having higher overall redox throughput through the Moore-Burn mechanism. We can say that while lactate is being produced, it can also go into mitochondria and become pyruvate. It can also, in the cytosol there can be some Moore-Burn happening if ROS is available. Cancer cells historically have more ROS than normal cells. And that is why the rerouting, it's not just, if you see Warburg effect, what is Warburg effect? Even when oxygen tension is high, you see that cancer cells are able to respire and they make more lactate because there is upregulation of LDH genes and there is more lactate production. But that lactate cannot enter the mitochondria too much. It is limited. So it has to go out into the bloodstream and go into the liver. So now, metabolic rates are higher in cancer, ROS is higher in cancer, which can support Moore-Burn based conversion. High cytoplasmic Moore-Burn is there, and then there is, I hope, a way which we can discover where we can burn out cancer cells using Moore-Burn principles and kill them rather than allowing them to proliferate. Now, I've already discussed this slide. There is some data here. So, Abhinav and we were discussing, and his PhD scholar did this work, credit to her, Nadia, Sheikh Nadia, and Abhinav is the guide. So here I want to explain this to you very quickly. You see, we showed that experimentally lactate cannot be reconverted to pyruvate. So you can see there is B, that is buffer, P is pyruvate, NADH. So what are we monitoring here? The rate per molar per second that you see is we are monitoring the NADH in the system, right? NADH, when you have pyruvate, what is happening to NADH? NADH is being consumed. So here you can see that, at different pH, you have both LLDH and DLDH. Two different forms of LDH were used. DLDH is bacterial LDH, LLDH is your normal, lactate dehydrogenase. You can see that at pH 7.4, the rate is very, very high. And at pH 9 also it is very high. So pyruvate is converted to lactate very readily, and you can see the y-axis, the numbers are quite high. But if you see this, here, you can see when we are checking lactate to pyruvate reconversion. Right here, what should happen if lactate is reconverted to pyruvate? NAD plus should become NADH plus H plus. The formation of NADH plus H plus is not very feasible. You can see the numbers are very low, and it is happening at pH 7.4 and 9, but the numbers are very low. It was 2000 odd when you saw the pyruvate to lactate reaction. That is NADH utilization. NADH formation is not so straightforward in our system because H plus formation is not thermodynamically very feasible according to the calculations that were done. Now if you see, we go on, liver tissue was taken, liver lysate, and then NAD plus was added to check for NADH formation. And you have lactic acid, La is lactic acid, LT is liver tissue lysate, B is buffer, and NAD plus was added, which means it should become NADH, right? If you add LLDH and DLDH, both of them are doing something amazing. They are messing up with NADH formation. NADH formation is going down. If you take liver tissue alone, NADH formation is more. You can see the pink bars to the left. But when you add LDH, the moment you add LDH, NADH is disappearing. So LDH consumes NADH rather than forms it. Okay. So that is one story. And again, this analysis was done with KO2, which is potassium superoxide. The moment you add potassium superoxide in the reaction, and you can see that there is conversion of, NADH formation is there, NADH formation, which means that lactate is becoming pyruvate. But if you add LDH, the values go down significantly. Now, FTIR analysis was done to see the changes in spectra between lactate and pyruvate, and then you can see that lactate is not getting reconverted to pyruvate. GCMS was done to back up this observation using superoxide catalyzed oxidation of lactate, and Abhinav could show that superoxide itself could do it, could convert lactate to pyruvate. But the moment you add LDH, right, you can see here LDH, if you add, it's going down. So LDH is messing with the system. That's why LDH is able to do pyruvate to lactate and not lactate to pyruvate. That's something that is very clear from the data. So you can see here in the GCMS analysis that superoxide can do it in the control reaction. You have both lactate and pyruvate at the same level, but then when you add KO2, right, you are seeing that lactate is getting consumed by KO2. KO2 itself can do it. That is support for liver being able to convert lactate to pyruvate. Okay, so now I am moving on to the next system. If you have questions, you can ask me later. I have very less time. I'll try to finish in 10 or so minutes. So the other system is cyclooxygenase. Cyclooxygenase is very famous for what? Pain, right? The pain and inflammation is because of activity of cyclooxygenase. The natural substrate of cyclooxygenase is arachidonic acid, icosahexatetraenoic acid. We tried to reinterpret the structure function correlations of mammalian cyclooxygenase using Morvan concept. This is cyclooxygenase. You can see there, right, the active site channel for entry of arachidonic acid is very, very small. Certain crystal structures show that this can open up a bit more. But I'll come to the details. It is also a glycosylated enzyme, right? And it is monotopically inserted. Monotopically inserted means it is loosely bound to membranes. Cyclooxygenase is there near membranes. LDH is a soluble murzyme, but cyclooxygenase is more a membrane-associated murzyme. That's something that I should tell you now. So, what does cyclooxygenase do? It takes arachidonic acid in 2 steps, in 2 different active sites. Cyclooxygenase has 2 different active sites. One is called the peroxidase site and the other one is called the cyclooxygenase site. Both of them are facing opposite to each other. The cyclooxygenase site is into the membrane. The peroxidase site is on the opposite side, away from the membrane and exposed to the solvent. So now you have interfacial chemistry here because membrane is there, solvent is there, the enzyme is able to do both, right? Wonderful enzyme. So you have arachidonic acid at a particular carbon 13, it gets attacked and there is endoperoxide formation, right? You have something known as PGG2. PGG2 is actually the product of cyclooxygenase site. It goes to the peroxidase site and then it becomes PGH2. So if this has to happen probabilistically, Murali sir did a calculation. taking all the steps and the probability considerations, and for this to happen according to the classical way, it is a probability of 1 divided by 576. That is very difficult. So, because you need this thing which goes in like noodles, right? It comes out like a hairpin, and we don't know how feasible this binding and dissociation is. We have not studied that yet. Later on, what happens to prostaglandin is you get prostacyclin, thromboxane, all these are involved in inflammation, right? Inflammatory mediators. That is why NSAIDs can block cyclooxygenase activity and then stop the process of inflammation. That's why we have NSAIDs.
Yes, Laurent Gideon.
Now, I don't want to go into this, already explained the classical mechanism, because there are so many steps. That's why these calculations were done, and that is a low probability thing for such a fruitful association to occur. Active site. Now, people say that, there are slight differences in the active site volumes of cyclooxygenase 1 and cyclooxygenase 2. Cyclooxygenase 1 is always seen in your system. Cyclooxygenase 2 is inducible. Whenever you have some inflammation or ROS or oxidative stress, cyclooxygenase 2 is expressed and made. So the minor changes in the active site volume, they say, leads to the variability in substrate and inhibitor preferences. So therefore there are COX-1 specific inhibitors, COX-2 specific inhibitors, and dual inhibitors of both the isozymes. So we tried to study that in both the peroxidase and cyclooxygenase site. We bound the molecules and checked whether there is any scope for differentiating the substrates of arachidonic acid, prostaglandin H2 and PGG2. Now you must understand that PGG2 is formed first, arachidonic acid becomes PGG2 in the cyclooxygenase site. PGG2 has to go the other way around. What is the dynamics involved? Arachidonic acid has to come out of the membrane and go into cyclooxygenase site. That has to become PGG2. PGG2 has to come out of the membrane and go to the solvent, and from there it has to come that side. So it's a very intricate mechanism that is proposed by people. But these are all lipid molecules. How well would they just come out of lipid? Right? So there is a very interesting crystal structure of mouse cyclooxygenase which I found, in which there is both productive and non-productive binding of arachidonic acid. In one case, the COOH group is outside. The other part, the methyl end, goes in. In the other one, the orientation is ulta. In the first case, A, they are saying that it is the correct or productive orientation. The other one they are saying is the non-productive or allosteric orientation. So, there was one paper where they showed that all these molecules here, that cyclooxygenase is a dimer. Okay, understand that cyclooxygenase is a dimer. So, there are two copies bound to each other. So, they are saying that one is an allosteric site, the other one is a catalytic site. This also didn't make much sense to me. Why one should be allosteric and the other one catalytic? Different substrates have different substrate binding and preference for both cyclooxygenase 1 and 2. You can see according to the color code, there are efficient cyclooxygenase substrates, allosteric stimulators, inefficient COX substrates which are competing with arachidonic acid, and allosteric inhibitors. All this looks like a very elaborate story rather than, grounded in simple explanations. So we did a lot of docking of different compounds. I'll just tell you, I'm not going into the details, I'm just going to tell you that cyclooxygenase 1 specific inhibitors were found to bind to even COX-2. COX-2 specific inhibitors were able to bind to COX-1. That's why you cannot have a concept of specificity. Because these inhibitors can go into both active sites. You don't have I think it is a myth of saying COX-1 specific and COX-2 specific. That's why in medicine, cyclooxygenase inhibitors, if you take COX-2 inhibitors, they can also bind to COX-1 and kill you. Right? There is that cross specificity. There is no specific binding. How can we explain that? When we saw the protein, we saw that there are arachidonic acid entry channels. Tyrosine 385 is the most important residue in the active site, very deep active site of 25-30 angstrom. Tyrosine 385 is there and arachidonic acid squirms in and then it is converted to PGG2 and it comes out. There are so many channels. What is the role of all these channels? That is a question that nobody has answered. Why will it use only one channel? Why not other channels? Now coming to the last slide perhaps, I'll show you some mercy and leave you. So, in the Morburn view, Ross generation is always there in cyclooxygenase. You take any literature, you will see singlet oxygen, superoxide, hydroxyl radical. We say that all the NSAIDs you can see here in the right, they can mess with the DROS which is available rather than having this thing going in and then something coming out and that going there and getting converted into something else. We are saying that it's a very simple thing that ROS which is there comes out into the lipid and it can convert. So that is what these equations are saying. Because of lack of time, I am not going to explain that. We are saying that ROS can do it. ROS is good. That's all. I conclude. I'll be happy to take questions.
Sir, you have shown cancer cells, they are not going through Warburg effect.
They are going through Warburg.
Yeah. So
Warburg effect, what do you mean by Warburg effect? Even in high O2 tension.
one doubt which I have, you are also telling that ROS can be the reason apart from the Warburg effect. That's what I am.
Exactly. No, not apart from Warburg effect. I think you misunderstood. What is Warburg effect? It is there in cancer. What do cancer cells have? They usually have more ROS than normal cells. Right? That's where the Morburn concept can apply. Normal cells, they cannot reconvert lactate to pyruvate. See, if you are taking a sprint, let's say you are running a 100-meter dash. By the time you finish, you get pain in your muscles. Why do you get pain? Because of lactate accumulation. Okay, now that lactate cannot be normally reconverted. It has to go into the mitochondria. Interestingly, in humans, mitochondrial LDH is a DLDH. It is not a LLDH. Okay, so you know that D and LLDH will operate differently, and also that DLDH is a flavozyme. It's a very different system. So in the mitochondria, why does it go there? Simply in the Morburn view, mitochondria is rich in ROS. People have calculated that around 10 million electronically modified oxygen derivatives are produced per mitochondrion per day. So therefore, being rich in ROS, mitochondria can reconvert. But how much of lactate can go into mitochondria? That is limited.
Sir,
right?
Experimentally this is not much proven, right? Because in world, if you take millions of people they have done PET scanning, positron emission tomography. Because if you have one brain tumor,
I'm not disagreeing with Warburg. I'm not saying Warburg is not happening.
I understand that it can be an alternative.
I think you have misunderstood. You can talk to me and do my best to explain.
Yeah, so once again, let us thank both the speakers for the wonderful session. Thanks to all the organizers for this wonderful opportunity. Thank you.
Thank you, madam. Thank you so much. I just wanted to say one comment to my student Danny. Cerebrum P50 and reductase, these puppies, I really like that. Once you start working with some proteins, you start loving them. You start liking them. You start humanizing them. You start animating them. So I really like that. Nice, sir. Yes, yeah, Lauren Jenkins, can we just have Lauren Jenkins give a message? Just a message. He was supposed to join yesterday, there was some problem, and today he has got a message. Can we read out what, first, I'll read out, do you have Lauren Jenkins' CV? Do you have Lauren?